Welcome back to this educational channel called Learn Bio with Janet. I'm teacher Janet. Today we'll be studying the topic of variation, which is a continuation from the first video. And this time we'll be studying subtopic 6.2, causes of variation, which includes crossing over, random fertilization, and mutation. So let's begin. The learning outcomes for this lesson are as follows. After this lesson, you should be able to state the factors causing variation. For example, the genetic factors and the environmental factors. Secondly, you should be able to explain the effects of genetic factors on variation. For example, the effects of crossing over on variation. And thirdly, you should be able to explain mutation by giving the definition, the causes, and then examples of mutation. Firstly, let's look at this overview of the causes of variation. So variation can be caused by genetic factors or environmental factors. Now, for genetic factors, the genetic factors that cause variation are the events involved in sexual reproduction. For example, in the formation of gametes, during meiosis 1, there's crossing over and independent assortment of homologous chromosomes. And then there is random fertilization in the process of sexual reproduction. So these three events will produce a new combinations of genes in the individuals. Fourthly, there is one more factor, which is mutation, that can produce new genes which can be inherited. Then for environmental factors, which you will study in another video, examples are as follows: sunlight, nutrition, and temperature. Okay, for example, exposure to sunlight can increase the pigmentation on the skin and make it darker. Let us start with the first genetic factor that causes variation. This is crossing over, which occurs during prophase 1 of meiosis in the formation of gametes. So if you are asked when crossing over occurs, just remember that crossing over occurs during the first phase of meiosis because it is one of the most important processes that occur in meiosis. So it occurs early on during the first phase of meiosis, which is prophase 1. Now, what is crossing over? It is actually the exchange of genetic material between non-sister chromatids of a pair of homologous chromosomes. So if we are asked to explain the crossing over in detail, let's read from here. The non-sister chromatids of homologous chromosomes will break at the chiasma, which is the point of crossing over. Now, homologous chromosomes are chromosomes that are of the same type, and they have the same structure and the same shape, and they carry the same type of genes. So one is from the father, is the paternal chromosome, and the other copy is from the mother and is called the maternal chromosome. For example, paternal chromosome number 1 in humans and the maternal chromosome number 1 in humans. This will make up a pair of homologous chromosomes. So non-sister chromatids means one chromatid from the paternal chromosome and another chromatid which is from the other chromosome, which is the maternal chromosome. So these two chromatids are called non-sister chromatids. They touch each other and then they break at the chiasma, which is the point of crossing over. Next, they will exchange segments of chromatids between the paternal and maternal chromosomes. There's exchange of the segments of chromatids. So we say that there's exchange of genetic material between the paternal and maternal chromosomes to produce new combinations of genes. All right? So sometimes this is called genetic recombination, new combination of genes on both the paternal and maternal chromosomes. So at the end of meiosis, it will produce genetically different gametes, even though these gametes are from the same individual. In this way, it will cause variation, even variation in between individuals of the same family. Here is a diagram that illustrates very well how crossing over causes new combination of genes to be found in the gametes. Here is a paternal chromosome, and here is a maternal chromosome. So this is a pair of homologous chromosomes. And let's say this man is producing the sperms, all right? So his father has contributed this chromosome to him, and the mother contributed this chromosome. Let's say it's chromosome number 1 in the human karyotype, right? So the father's chromosome contains or carries some dominant alleles, which is a version of a gene. And then the mother's chromosome contains or carries some recessive alleles. All right. For example, maybe father has black hair and mother has yellow hair, as an example. Okay, which is a yellow hair allele is a recessive allele. Black hair allele is a dominant allele. So maybe for blood group, father has blood group A, and he has the allele for blood group A. He carries the allele for blood group A, but mother has the recessive allele for blood group, which is the allele for blood group O, IO. So this is how they have different versions of the genes, different versions of a gene. So during crossing over, the chromosomes or sister chromatids, non-sister chromatids, will overlap. So one chromatid from the father overlaps with one chromatid from the mother at the point of crossing over called the chiasma. So here. All right, then the chromatids will break off, the segments will break off and rejoin to the other chromosome, to a different chromosome. So now father's chromatid contains a segment of the mother's chromatid with the recessive alleles. And mother's chromatid here contains a segment of the father's chromatid with dominant alleles. So these two are new combinations of genes. Then when meiosis is over, these sister chromatids would have separated from this one, and this one from this one. And they will move to different poles, and they'll be found in different gametes. So the first gamete will have the original versions of the genes from the father of this man, and that is all the dominant alleles. And these are all the dominant alleles. But the second one, this one, it has new combination of genes, right? So it has a dominant allele here, capital big A, and then small b and small d. And the third one, the maternal chromosome or chromatid, has one recessive allele and two dominant alleles from the father. So this is a new combination of alleles or genes. Okay, and this one is based on the mother's collection of alleles. It's still the same, like the original recipe in inverted commas. So here we see two new combinations of alleles as an example of how there is genetic recombination. So all these four gametes are genetically different from each other, and is produced because of crossing over. It occurs because of crossing over. Okay? So these gametes are going to be used to produce the next generation, which is the the offspring of this man. Alright, so we see that the genes from the father and mother are passed down from one generation to the next and to the grandchildren in this case. Alright, because if any of these gametes were to undergo fertilization, then it will lead to the formation of a new offspring. The second genetic factor that causes variation is the independent assortment of homologous chromosomes during metaphase 1. So during metaphase 1 of meiosis, the homologous pairs of chromosomes, each consisting of a paternal chromosome and a maternal chromosome, are arranged on the metaphase plate at random. Or we say that there is random arrangement of homologous chromosomes at the metaphase plate, meaning the equator or equatorial plane of the cell, in the center of the cell. So each pair of homologous chromosomes is placed or positioned relative to the poles of the cell independently compared to the other pairs. They don't affect each other, the different pairs of homologous chromosomes. The way they are arranged at the metaphase plate, that means facing which pole, the left or right pole for example, is at random, occurs randomly. Randomly means secara rawak, or in Chinese we say 随便. There's no fixed pattern to it. Thus this produces different combinations of paternal and maternal chromosomes in the gametes. And so the gametes are genetically different from each other. Let's have a look at the example given for this in the next slide. Let us discuss an example to show that independent assortment of homologous chromosomes in metaphase during metaphase 1 causes variation. So actually this event causes variation through the formation of genetically different gametes which have different combinations of paternal and maternal chromosomes. Different combinations of paternal and maternal chromosomes. Now let's take a cell where 2n equals to 4. So this is a parent diploid cell. Imagine that is undergoing meiosis to form the gametes. So how does it form genetically different gametes? So in this cell there are 2 pairs of homologous chromosomes. 2n equals to 4. So there are 2 pairs of homologous chromosomes, and in each pair there is a maternal chromosome represented by the blue chromosome and a paternal chromosome represented by the red chromosomes. Okay, then the 2 types of chromosome, the long chromosome and the short chromosome, are different from each other. And also if we compare the maternal chromosome with the paternal chromosome in a pair of homologous chromosomes, they carry different alleles or versions of genes. They carry different versions of genes. Maybe mother has the allele for brown hair, but father has the allele for yellow hair. Alleles are different versions of the genes. Mother may have the allele for blood group A, father may have the allele for blood group B. Okay, so during metaphase 1 in meiosis, homologous chromosomes will be arranged at the metaphase plate in pairs. And there are 2 possible arrangements, okay, for this event. In the first possible case, the maternal chromosomes, long maternal chromosome and short maternal chromosome, may face the same pole, and the 2 paternal chromosomes may face the other pole. Okay, so in anaphase 1, the homologous chromosomes will separate from each other and move to opposite poles. So these 2 chromosomes will be found together in the same daughter cell after meiosis 1. And the paternal chromosomes will be together in the same cell after meiosis 1. Now in meiosis 2, during the metaphase 2, the chromosomes will be arranged at the metaphase plate, and then in anaphase 2, the sister chromatids will separate from each other and move to opposite poles. Okay, so here we see the final result. This is a type of gamete form with the first combination of paternal and maternal chromosomes, but here we don't have any paternal chromosomes, only maternal chromosomes in this gamete. Then for the second daughter cell, it will also produce two gametes, but they are just the paternal. They only have the paternal chromosomes in them. Okay, so these two combinations of chromosomes are different. Now, what are the other possible combinations of paternal and maternal chromosomes? So now we have to look at another possible arrangement of chromosomes at the metaphase plate during metaphase 1. Okay, so in this case, the blue maternal chromosome faces the same pole as the red paternal chromosome. The long maternal chromosome faces the same pole as the short paternal chromosome, and the long paternal chromosome faces the same pole as the short maternal chromosome. Okay, so following the same process, we will find that these two chromosomes will come together in the same cell, and then in anaphase 2, they will the sister chromatids will separate, and finally you get the third combination of paternal and maternal chromosomes, or the third genetic composition in the cell, and this is the fourth. Okay, now this combination is unlike this combination, right? So here we have four genetically different gametes formed from the different arrangements of paternal and maternal chromosomes. So this is how the independent assortment of homologous chromosomes in metaphase 1 causes the formation of genetically different gametes, and this leads to variation. Here's an interesting fact that may help us to understand better how the independent assortment of homologous chromosomes during metaphase 1 can cause variation. So we know that in humans, the 2n number or diploid chromosomal number is 46. Now, what is the number of genetically different human gametes that can be formed in an individual based on the different combinations of paternal and maternal chromosomes? So we can use the formula 2 to the power of n, where n stands for the haploid number of the organism. For humans, the haploid number n equals to 23. So the number of genetically different human gametes that can be formed based on the different combinations of paternal and maternal chromosomes in an individual equals to 2 to the power of 23. And using the calculator, we can find out that this equals to 8,388,608 genetically different gametes that can be formed in an individual based on the different combinations of paternal and maternal chromosomes. This is actually a maths problem, right? Now, this is not even considering crossing over. So if we consider crossing over, it's quite possible that there are no two gametes that are exactly alike genetically, even though they are from the same individual. The third event that causes variation is called random fertilization. So basically it means that during the process of fertilization, any of the male gametes present may fuse with the female gamete present. It occurs randomly and there's no fixed pattern. So each gamete has a unique combination of genes, meaning that each gamete is genetically different from the other. Okay, each gamete, each sperm or each ovum is genetically different from the other, although they may be from the same individual, produced in the same individual. So in the female reproductive system, during fertilization, the fertilization between a male gamete, that means the sperm, and a female gamete or ovum, occurs randomly. So an ovum can be fertilized by any of the sperms out of the millions that are present. Just depends on which sperm is able to penetrate the membrane of the ovum the fastest and be able to have its nucleus fused with the nucleus of the ovum. Okay, so there's only one winner. All right, the first sperm that enters is the one that fuses, and the rest will not be able to enter after that. So we can say that basically each one of us is a winner because we have won the first race that led to our existence in this world, right? Now, thus each zygote produced is unique and genetically different from other zygotes. All right, so all of us are unique in that sense that we have a certain collection of genes, unique collection of genes that nobody else has, unless we have identical twin. We have an identical twin. The fourth genetic factor that causes variation is mutation. Now, mutation is a sudden random change in the genetic composition or material of a cell. By genetic composition or material, we mean the chromosomal structure or number or the DNA. That means changes occur to the chromosomes or DNA due to exposure to some agents of mutation. Okay, so it leads to changes in the genotype, and then from the changes in genotype, it will affect the characteristics or phenotype of the organism. So mutation is permanent and irreversible, and mutation can occur in somatic cells or normal body cells. This will cause uncontrolled mitosis leading to cancer. For example, if a person is exposed to X-rays or excessive ultraviolet light, so these rays can damage the DNA that controls the cell cycle. As a result, uncontrolled mitosis will occur because the genes that control the cell cycle are damaged. So mitosis occurs uncontrollably, leading to the formation of tumors and cancer. Now, mutation can also occur in gametes, in the formation of gametes. This will cause genetic diseases like Down syndrome and other hereditary diseases that like albinism and hemophilia that can be passed down to the offspring. Now, what causes mutation? So mutation is caused by mutagens or agents of mutation, such as radiation, for example, X-rays, ultraviolet rays from the sun, gamma rays, nuclear radiation. Okay, for example, X-rays. So we should limit our exposure to X-rays because it can cause mutation. And then for the sun's UV rays, which can be very dangerous, we should also make sure that we are not exposed too much to the rays of the sun. Now, chemical mutagens include benzene, formaldehyde, tar, cigarette smoke, pesticides, food preservatives. For example, the tar in cigarette smoke has been known to cause lung cancer. And then food preservative includes those that are used to preserve food like meats, okay, the nitrites that are used to preserve meat and sausages and so forth that we eat in our food. So we should limit our intake of this type of foods and don't eat it excessively. Now, what are the effects of the mutagens? So they cause mutations or changes such as mistakes that occur during the DNA replication in the S phase of the cell, during the interphase period of the cell, during S phase when DNA is being replicated or the chromosome is being duplicated, mistakes can be made if a person is exposed to the mutagens. And then during meiosis 1, especially during anaphase 1, there may be uneven distribution of chromosomes during the anaphase 1 of meiosis when the gametes are being formed. So we're going to discuss this in a short while, and this is called non-disjunction. As we have said just now, mutation involves changes in the genetic composition or material of the cell. So there are two types of mutation. The first type involves the chromosomes, and it is called chromosomal mutation. The second type involves the DNA, and it is called gene mutation. So in chromosomal mutation, for chromosomal mutation, there are two types of changes. Firstly, change in the number of chromosomes that lead to formation of Down syndrome patient or Klinefelter's syndrome or Turner's syndrome patient. And the second type of chromosomal mutation involves changes in the structure of chromosomes caused by deletion occurring in the chromosomes, duplication, inversion, and translocation. Gene mutations. For gene mutations, there are changes in the base sequence of DNA, meaning in the sequence of the bases such as adenine, thymine, cytosine, and guanine. ATCG. So when there is a change in the normal base sequence of DNA, this can cause the type of protein formed to be different, because changes in base sequence will lead to changes in the type of amino acids that are used to build the protein molecule. So it may produce a protein that is not able to function in the cell. So changes in the base sequence are caused by deletion, insertion, substitution, inversion, and duplication. Now, B and C will be studied in the next video. For this video, we'll concentrate on A, changes in the number of chromosomes. So here are the notes for the schematic diagram just now. Two types of mutations. Chromosomal mutations involve changes to the number of chromosomes, such as Down syndrome and Klinefelter's syndrome. And secondly, it also involves changes in the structure of the chromosomes, and it's caused by deletion, inversion, duplication, translocation. Short form is DIDT. Okay, DIDT. Now, as for gene mutations, it involves changes in the nucleotide or base sequence of a gene. So examples are when there is, we also call it deletion, inversion, duplication, insertion, and substitution, involving the bases, adenine, thymine, guanine, and cytosine. So examples of diseases caused by gene mutations are sickle cell anemia, thalassemia, albinism, and hemophilia. So all these are serious, many of these are serious diseases. They are caused by gene mutations. Let us now discuss the first type of mutation, which is a type of chromosomal mutation. This involves changes in the number of chromosomes in the cell. So a change in the number of chromosomes can take place when homologous chromosomes fail to separate in anaphase 1 of meiosis during the formation of gametes. And this is called non-disjunction. So non-disjunction means that is a process whereby homologous chromosomes fail to separate and move to opposite poles during the formation of gametes, for example. So both the chromosomes will move to the same pole and be found in the same cell. So this is an abnormal condition. Now this will result in, finally when meiosis is over, it will result in one daughter cell or gamete having an extra chromosome, the haploid number plus 1. For humans, this is 23 plus 1 equals to 24 chromosomes, while the other daughter cell or gamete has one less chromosome, which is 23 minus 1. That means only 22 chromosomes. So a normal gamete, human gamete, should have 23 chromosomes. Okay, but because of non-disjunction, the abnormal gamete formed has 24 chromosomes or 22 chromosomes. So there's addition or loss of chromosomes in the cells or in the gametes formed. Now this will give rise to genetic diseases. That means if the abnormal gamete were to undergo fertilization and fuse with a normal gamete, it can produce a condition like Down syndrome, Turner syndrome, or Klinefelter's syndrome. Now for Down syndrome, the number of chromosomes equals to 45 plus XX for girls or 45 plus XY for boys, and is sometimes called trisomy 21, meaning tri means 3, somy for chromosomes. So 3 chromosomes number 21. We'll discuss this in another slide. For Turner syndrome, the number of chromosomes is 44 plus X. Sometimes it's written as 44 plus X 0. That means lacking one sex chromosome. So for Klinefelter syndrome, the number of chromosomes is 44 plus XXY. That means there's an extra sex chromosome. That is, the extra chromosome is the X chromosome. Instead of having one X chromosome, there are 2 X chromosomes. So altogether, there are 3 sex chromosomes instead of 2. Now, how does non-disjunction lead to mutation? So let's take an example of a Down syndrome child. How is a Down syndrome child formed? So in non-disjunction, the homologous chromosomes fail to separate and move to opposite poles. Let's take the example of the Down syndrome child. Okay, so during the formation of gametes in the Down syndrome child's parent, one of the parents, either the father or mother, during the formation of the gametes, the homologous chromosomes number 21 fail to separate from each other. So both of the chromosomes 21 move to the same pole and will be found in the same daughter cell after anaphase 1, after meiosis 1. Then in anaphase 2, these 3 chromosomes are together. So in anaphase 2, their sister chromatids will separate from each other. This will lead to the formation of gametes that are abnormal, having one extra chromosome number 21. So a gamete should only have one chromosome 21 and one copy of the other chromosomes. But here you have 2 copies of the chromosome number 21 in the gamete. So this is an abnormal gamete with an extra chromosome, and that is chromosome 21. So if this gamete were to undergo fertilization with a normal gamete, with another normal gamete, it will form a zygote with an extra chromosome, and that is chromosome number 21. Well, there will be 3 chromosome 21 in the zygote. Okay. Now, whereas for this other cell that's formed, these other gametes that are formed, these 2, they will lack the chromosome number 21. So the number of chromosomes in the gamete will be n-1, which is 23 minus 1, becoming 22. Okay, for Down syndrome child, the gamete that leads to the formation of the Down syndrome child, one of the gametes will have 24 chromosomes, n plus 1, 23 plus 1 equals to 24 chromosomes, and the other gamete is normal. The other gamete that fuses with this gamete is normal. So 24 plus 23 becomes 47 chromosomes. Let's discuss 3 examples of chromosomal mutations where there is a change in the number of chromosomes. Now, firstly, we have to know that for a normal person, the total number of chromosomes in a diploid cell, or the diploid chromosomal number, is 46. And if we break it down, it is 44 autosomes plus 2 sex chromosomes. So autosomes are chromosomes that do not determine the sex of the organism. Alright? So a normal person has either 44 plus XY chromosomes. That means the total number of chromosomes equals to 44 plus XY for males. And for females, it is 44 plus XX. Okay, now we look at the chromosome mutation. Let's say we look at Down syndrome individual, which can either be male or female. So in total, they have 47 chromosomes. And if we break down the autosomes, divide them into autosomes and sex chromosomes, there are 45 autosomes and 2 sex chromosomes. So the sex chromosomes are normal. For males, the sex chromosomes are XY, and for females, the sex chromosomes are XX, 2 X chromosomes. So let's look at the autosomes. Compared to a normal person, there's one extra chromosome. And that extra chromosome is an extra copy of chromosome number 21. So in a normal person, there are 2 copies of chromosome number 21 in each diploid cell. But for a Down syndrome individual, there are 3 copies of the chromosome number 21. Okay, 3 copies of the chromosome number 21 in each of the diploid cells. So the symptoms for Down syndrome, we have to know at least 3, right? You can be asked to list or state 3 symptoms, for example. Symptoms are flat, broad face. Down syndrome individual will have flat, broad face, slanted eyes, protruding tongue. So it has been found that the tongue has poor muscle tone, and also certain part of it is larger than normal. And also the mouth may be smaller than normal. So lastly, mentally retarded. And one more symptom is also infertile, although not all are infertile. So the degree to which they suffer from all these symptoms depends on the individual. Okay, so now let's look at Turner syndrome. For Turner syndrome, the individual has 45 chromosomes. And if we break it down, the total number of chromosomes is 44 plus X. So the autosomes are normal. It is the sex chromosome, all right, that is absent. One sex chromosome is absent. So a Turner syndrome individual is actually a female because she has no Y chromosome. Okay, a female with one X chromosome only and missing one X chromosome. Symptoms. This person who suffers from Turner syndrome is infertile, meaning she cannot reproduce or bear or have children. And she has underdeveloped breasts and ovaries because she's lacking one X chromosome. The last mutation we are studying here, the third one, is Klinefelter's syndrome. So in a person with Klinefelter syndrome, each cell has 47 chromosomes. That is 44 autosomes and 3 sex chromosomes, X, X, Y. So this individual is a male because of the Y chromosome. All right? Males have the Y chromosome. However, there's an extra X chromosome here. Okay? Thus, let's look at the symptoms. This person who suffers from Klinefelter syndrome is infertile, shows breast development because of the extra X chromosome, the influence of the extra X chromosome, and usually is mentally retarded. So for these 3 mutations, the cause is the same. and that is non-disjunction or homologous chromosomes fail to separate during meiosis. Now let us try to identify the karyotype for all these individuals which have chromosomal mutations. Here we see a karyotype, which is a photograph of a person's chromosomes arranged as pairs of homologous chromosomes in descending order of size from the largest to the smallest, and the last pair is a pair of sex chromosomes. So firstly, we have to identify whether this person is a male or female. Now, having two long X chromosomes, it means that the person is a female. Two X chromosomes. For males, they have one X chromosome which is longer and a Y chromosome which is shorter. Then next, the abnormality is in the number of chromosomes here, which is chromosome number 21. There are three chromosomes, chromosome number 21, three copies of chromosome number 21 instead of two. Okay, so circle it up. And this is this causes the Down syndrome to occur. Extra chromosome is chromosome number 21. So the total number of chromosomes is written as 45 plus XX for girls, or 45 plus XY for boys. Now let's look at this karyotype. What do you think about this karyotype? Is it a normal karyotype, or is it a karyotype that's abnormal? Now, if it's an abnormal karyotype, where is the abnormality and what is the condition called? Yes, so this condition, this karyotype is abnormal because there's only one sex chromosome, and this is the X chromosome. So this karyotype is that of an individual with Turner syndrome, okay, where there are 44 autosomes here and one sex chromosome only. So the number of chromosomes is 44 plus XO or X0, meaning that there's lacking one sex chromosome. How about this karyotype? Is it a normal karyotype? Not exactly, right? So the abnormality again is in the sex chromosomes. Instead of having two sex chromosomes, there are three. Okay, so this syndrome is called Klinefelter syndrome because there are two X chromosomes and the short one is the Y chromosome. So in Klinefelter syndrome, the patient has 44 autosomes and the three sex chromosomes, X, XY, in each cell. Lastly, here are three questions that you can try to apply what you have learned, to assess yourself, and also to practice doing the HOTS questions. All right, just to train your brain. So try these questions before you look at the answers. BioBrain Jeep. Now state three genetic factors that cause variation. Number 2, explain the cause of Down syndrome in a child, for marks. Number 3, explain how Turner syndrome occurs. So 2 and 3 are like HOTS questions, all right? Then after that, see the answers in the description box right below the video. That's the end of the lesson. So in the next lesson, we'll continue to discuss mutation part 2. And in the next video, we'll be discussing chromosomal mutation, where there are changes in the structure of the chromosomes. We'll also discuss gene mutation, where there are changes in the base sequence of the DNA or the gene. So thanks for watching. Stay tuned for the next video. Please share, like, and subscribe. Goodbye for now.